So you can see Dr. Zeb Tonkin on screen now, who's a senior scientist in Applied Aquatic Ecology at ARI. And Zeb's going to be talking about his extensive experience in the recruitment dynamics of freshwater fish, environmental flows, fish migratory behaviour, floodplain assemblages, threatened and pest species and habitat restoration. It's all jam-packed. So Zeb will be discussing his work on the monitoring ecology outcomes of water for the environment across Victoria. I'll hand over you to Zeb. Thanks.
Okay, thanks, Michelle, and thanks, Fern, both for the welcome to country and that introduction. And thanks, everyone, for dialing in today. So, as the title indicates, today I'll be giving a really high-level overview on the work ARI does in monitoring the ecological outcomes of water for the environment or environmental flows across Victoria. I'd like to acknowledge all the co-authors and collaborators who I'll mention throughout this talk, and also at the end of the talk, who have played huge roles in producing all this work that we've done in the past and continue to do. So I'll give a real high level overview of the problems, river regulation and environmental impacts, how we're trying to reduce these impacts using water for the environment, and the approaches and some results from DELP's key monitoring that ARI has been leading on the research monitoring to evaluate this intervention and importantly help us guide how we can do things better. So everything in this paper that I produced today is based on a suite of reports and journal articles that we've produced over the past few years. So if anyone needs any of these resources, please get in touch. So we're all aware of how river regulation has disrupted the way waterways flow naturally. We've disrupted the natural flow regime. We've reduced the frequency, magnitude and duration of winter and spring floods. We've altered low flow periods during summer, over-extracted water, or sometimes we put too much water into rivers during summer. And we've modified the natural variability that we see in our waterways. This has impacted connectivity, productivity, and the quantity and quality of critical habitat that subsequently has major impacts on ecosystems and native flora and fauna species. So to reduce the impacts of river regulation, we're increasingly turning to environmental flows or water for the environment. And this intervention aims to restore these aspects of the natural flow regime, or usually in these flow altered ecosystems. Environmental water is used to alter the frequency of inundation or the magnitude of flow events or even base flows. So we'll add water to a system to try and get some of that variability back, or we'll inundate floodplain habitats a bit more. We're doing this because of the key links there to functions or processes that govern populations or ecosystem outcomes. So we might put a pulse of water down the river to cue migration of a fish species. We might do that to enhance the dispersal of seeds, enhance connectivity, or simply to maintain water quality to keep aquatic fauna alive. But the success of this intervention, it really is reliant on having that great understanding between population processes or functions and that flow regime, but also accounting for other modifiers. So how do we do this? Through monitoring and research, of course. DELP's got a few key monitoring programs that it uses to do this. But one of the big ones, as the acronym suggests, is the Victorian Environmental Flow Monitoring Assessment Program, and more recently the wetland equivalent that kicked off in the mid-2000s. This monitoring and research program aims to test some of those flow recommendations and the way we deliver environmental water relative to those functional flow objectives or those processes, what we're trying to achieve with these parcels of water. The program also assesses long-term trends relative to that fundamental objective of enhancing populations or enhancing biodiversity. And it also conducts really targeted research to fill some of those knowledge gaps within our conceptual models that underpin these recommendations. So today I'll just touch on a few examples of each of those. So evaluating function and processes. So this has been one of the major changes to the monitoring program over the past 6 or 7 years. A lot of this function and process monitoring uses event-based sampling, And that's largely because environmental water is delivered as, these events, these freshes, a pulse of water is put down the river, as you can see in the diagram here. And as I mentioned before, due to the function and process, the objectives of those events, we're trying to, for something like riparian vegetation, we might be trying to enhance seed dispersal or recruitment or growth or survival. Similarly, for fish, for example, we're trying to enhance dispersal and migration queue, survival or a recruitment process. So this work's produced some really neat results. For fish in coastal systems like the Glenelg River, for example, we've seen up to a 6-fold increase in the numbers of diadromous fish. So these are species that need to move between marine or estuarine environments and up into freshwater reaches to complete their life cycle. So we've seen this 6-fold increase in some of our key species moving in response to these freshes delivered in summer and early autumn. Similarly, for some of our lowland systems up in the Murray-Darling Basin, we've seen these fishes increase tailwater levels at some of our key barriers, and this has subsequently enhanced the movement of the whole fish community past these barriers in lowland river reaches, places like the Loddon River, Gumbower Creek, and even in the Murray River itself. So the second approach that we're looking at monitoring is by looking at the whole communities. So yes, we might be successful in making a fish migrate, a seed sprout or a bird feed, but what does this mean in terms of communities and the whole of biodiversity, the whole of populations? It's much harder to tie back to specific flow events, but it's information that we need to gather regardless, just because of this fundamental objective of improving populations and improving biodiversity. So for vegetation, for example, we can look at metrics like diversity and foliage cover. And the work by Chris Jones here clearly shows the role environmental water this is in the Campaspe River, has done in maintaining the structure and foliage cover and fringing emergent vegetation of the fringing emergent vegetation community. That's really important, not just in terms of plant biodiversity or native plant biodiversity, but also the ecosystem function and what these plants do. So things like holding the bank together, reducing erosion, providing habitat for native fauna, that kind of thing. So some really neat stuff here, and this image really captures that nicely. Similarly, for something like fish, we undertake annual population surveys so we can track trends in the community composition and population dynamics through time. Here we can see in the Glenelg River, for example, for important diadromous species, just like those ones I mentioned before that we were trying to achieve event-based sort of outcomes for, there have been amazing outcomes with up to 16-fold increases in some species in population size since the Glenelg Hopkins CMA implemented some of these changes to the flow regime using environmental water back in 2017. We're also using tools that combine some of the learnings from the functional and population data to run some counterfactual assessments of our flow management outcomes, and importantly, make comparisons between different flow management scenarios. Here we've used population models for one of our long-lived fish species, Murray cod, which I'm sure many people are familiar with, in the Campaspe River, where we've parameterised the model construct with learnings we gained on the role of flow in governing survival and recruitment dynamics, and tested several flow scenarios. And this basically shows that the current flow recommendations produce a much larger population size and present a lower risk of crashing below a certain threshold in any given year. So again, a really useful tool. Finally, we can't always get the information we need from field monitoring, and some of the assumptions underpinning our conceptual models and population models, and indeed flow delivery, need to be tested via specific research questions. So a really neat recent example of this was some ex situ vegetation experiments conducted by RI's Lindsay Vivian and Chris Jones and some fantastic students from the University of Melbourne, Alana and Marjorie, to assess the role of temperature on inundation, or temperature from the inundation, to assess the role that temperature and inundation play on growth and survival of riparian vegetation. This work shows that unseasonal summer inundation that we sometimes see in our regulated systems that I mentioned earlier can have major negative impacts on survival due to the flowering stage of plants and high temperatures. Similar flows, however, delivered in winter and spring, like they should do under a natural flow regime, don't have the same impact on our native plants. And as a whole, this work's been really vital to understanding how timing of managed flows can influence vegetation structure. Of course, all this information is vital in the adaptive management loop. And like the other topics I've touched on, I could give a whole talk on this alone. But in short, the monitoring research information that some of the stuff I've mentioned before is used by managers in developing seasonal watering plans and subsequent water delivery. It provides evidence to continue the current recommendations, for example, provides data to refine our current recommendations and then subsequently test those refinements. And also it gives us evidence to avoid certain flow types, like some of the summer inundation stuff that I just mentioned previously. Researchers also contribute to environmental water advisory group meetings that happen annually for each of our key priority waterways across the state. And also all this information together, including just informal chats between researchers and waterway managers, provides feedback to CMAs to respond to key stakeholders like long-term trends in the abundance and distribution of key species at certain sites. Last but definitely not least, like a lot of the work we do at ARI, communications and engagement was identified in past reviews as being a really important part of the environmental flow monitoring program. Pam Clooney's been leading and has done in the past and continues to lead the charge on this, producing an incredible amount, diverse range and quality of products in showcasing this work, both from the researcher's perspective, as you can see here, which is only a fraction of the products that she's helped produce, but also importantly by supporting our partners like the Victorian Environmental Water Holder and all the catchment management authorities who are driving these interventions and basically just contributing to help them share the environmental watering story as a whole. Finally, all our monitoring and research also throws up really important information regarding key knowledge gaps. The Wet Map program, for example, showed that as a whole, providing water for the environment into wetlands across Victoria works in terms of making wetland vegetation go green or attracting common fish, frogs and birds, as you can see in this figure here. But as a whole, water for the environment is necessary, but not sufficient to restore complete function of wetland ecosystems. So common frog species show up quickly, but frog species of conservation concern, like the growling grass frog, don't necessarily recolonise. Birds use environmental water wetlands for feeding, but don't often breed. And in wetlands that have previously had several that have severely altered hydrology, environmental water helps common plants sort of re-establish, but herbaceous vegetation is nowhere near as species rich, and it's really much patchier than it should be. So for many taxa or sites, yes, environmental water is necessarily, but complementary interventions are also required. And this opens up new research questions or needs and also refines objectives with the tools that we've got at our disposal. So this leads us into what's ahead. So in summary, DELP's environmental flow monitoring program will continue to quantify functional and process responses to environmental flow delivery to help, refine some of that watering that we're talking about. It'll continue to monitor trends for key species or key systems to track progress towards improving that fundamental objective of improving populations or improving biodiversity. And this includes refining, just in the last sort of 6 to 12 months, refining a lot of our survey methods for some taxa or some sites to simplify assessments and also to standardise data across some of the other both state and Commonwealth monitoring programs, so we can share data, things like the GEO's MER program, Victoria's VMFRP program, and also MDBA's TLM Living Murray program. We'll also continue to that targeted research to fill those key knowledge gaps, and this includes those collaborations and links with students, which has been so valuable in the past. And we'll also continue to combine these data types and forecast long-term outcomes and compare management scenarios, which is really useful for the waterway managers on the ground. And finally, we'll continue to strengthen links with managers, policy communities and traditional owners. So a great recent example of this is the work Wayne Costa's doing with the Gunnakernai traditional owners looking at eel migration. And this has been a great example of bringing together those common grounds of environmental, water and cultural values. So finally, as mentioned at the start of the talk, I'd like to acknowledge the huge number of people and collaborators that have been part of this journey so far and will continue to be moving forward. And also, as mentioned, please get in touch if you're after any of the information on the work I've mentioned or anything that pops up if you're visiting the ARI website. So thanks.
Terrific. Thanks, Zeb. That was a really great run through, particularly environmental flows. And I found it quite interesting to note, I think in a lot of cases, we think that, you know, if you put the flows back, you know, that the rest will take care of itself. But it's interesting that it's actually brought up a whole stack of new questions, and particularly for threatened or species that are much more limited. So that was really valuable. There's a couple of questions there, and I know you did speak about interactions with managers about environmental flows, but there's a further question there from Deborah, who's asking about how ARI interacts with water holders and managers to ensure environmental flows or pressures, and whether DELP has any authority to require appropriate flows. Did you want to answer that one?
The first one, it's the first one's, you know, interaction questions. That's really diverse, I guess, and I guess it's a lot of it's really participatory action. That's some of the that's a big one, in that, you know, it can be as simple as a phone call to a waterway manager, and vice versa, saying, hey, we're delivering a fresh this week. We know you've got this monitoring in place. Let's see how we go. And it's you know, we'll meet up on site. We'll share results, you know, as they come to hand. So observations. Okay, 2 weeks time, we've got another one of these. How can we tweak things? We didn't see what we thought we'd see, or whatever it might be. And then there's the more formal sort of interactions in terms of reports or scientific papers that are sort of more medium term sort of interactions, in that they get embedded in flow studies or seasonal watering plans or whatever it might be. And then there's the much longer term scenarios, the EWOMP sort of stuff. So there's sort of different scales, that everything from a simple phone call or email that can happen on a daily basis, right up to that formal stuff that happens over 5-year periods. So it's a bit of a range.
And it's really important the science underpins the decision making by those land managers or those water authorities. So that's really helpful. There's a question from Andy about how your research links to studies by scientists in the Murray-Darling Basin, and how much collaboration is there in that space?
Yep, again, I sort of really briefly touched on that one in moving forward sort of space, but absolutely, there's, with so many monitoring programs going on in different areas, similar questions. It's been a really big focus, probably the last, I don't know, 5 to 10 years, and that we've got a lot of data sharing going on, a lot of collaborations underway. So the Commonwealth's Flow MER program, for example, we share data with the VEFMAP program. What are some other examples of this? There's a lot of the targeted research questions, for example. We get co-contributions from the different research programs, and let's say things like birds and fish, for example, don't sort of often don't recognise river boundaries or state boundaries. So we need to assess these populations at the scales that they actually operate, and we have to do these collaborations and share data if we want to answer them properly.
Yeah, I think that's a really good point. And Sandra's asking to elaborate, which I think is kind of related, on how you plan to get better standardisation of monitoring protocols and data management across agencies and jurisdictions. I think ARI plays a really important role in that space.
For sure. The standardisation one, I mean, there's standardisation in terms of monitoring approaches, which I sort of mentioned before, but there's also standardisations of flow metrics that as researchers we report on as well. So that's a tricky space, that one, but it's definitely something that's on the, you know, the forefront of our minds and the conversations that we have with managers.
Yeah, and it's about having that influence, isn't it, into other people's systems and organisations and approaches. So, and I think ARI's reputation in terms of expertise and the quality of the work probably has a lot of standing and influence in that space to be able to kind of provide an opportunity to get that standardisation.
Yeah, definitely.
And a colleague of yours, Brad, is asking about whether you'd like to comment on how technology might enhance or improve monitoring in future, such as the eDNA or the drones or those things.
Absolutely, and it already does make up a huge part. We use otolith microchemistry to, some pretty funky techniques to assess where a fish may have been born or where it's moved to. Definitely, we're currently using drones, or our collaborators use drones for things like bank erosion and vegetation assessments. And for sure, and a lot of the work that our collaborators at universities and students, they're sort of at the forefront of a lot of this as well. So that's really important to maintain those links, so we have the avenue to sort of explore some of that new technology.
Terrific. Well, thank you so much, Zeb, and for answering all of those questions as well. Really great presentation. So we're going to move now from Zeb, who's been sitting on Wurundjeri country, to our second presenter, Dr. Tracy Reagan, who's a principal research scientist sitting on Tanarung country. And Tracy is going to talk to us about her interests, which are very broad, encompassing